So last time when we finished up talking about the Calvin cycle, at the end of photosynthesis, we had mentioned that the actual end product of the Calvin cycle is 3 carbon sugars, or triose phosphates. And those are being made in the stroma of the chloroplast, and then they are moved into the cytoplasm through this exchanger protein that swaps a free phosphate for a triose phosphate. And then those 3 carbon sugars are in the cytoplasm, and they can be formed into sucrose. How many carbons was sucrose?
4 or 6.
6. Yeah. 6, similar to glucose. But sucrose is what the plant ships to non-photosynthetic tissues. And how fast a plant is making sucrose depends on how much CO2 assimilation is going on. And that word is going to come up in the next couple of lectures. And so you can think of CO2 assimilation as being the same thing as carbon fixation. So that's really talking about the same process. Getting carbon out of the air and stuck onto organic molecules. So you can call that carbon fixation, or you can call it CO2 assimilation. And so if the plant is doing a lot of photosynthesis, it tends to make more sucrose. But that's also balanced against how much sucrose is the rest of the plant using or not. So plant cells can sense how much sucrose is being siphoned off, how much CO2 is being fixed, and adjust how much sucrose it's making moment to moment in that fashion. So again, plants are making their own sugars. Some of this sucrose will actually be used within this cell as a fuel. The extra is shipped off to the rest of the plant. So some of the sugars that are made during photosynthesis are used for energy through aerobic respiration that we're learning about in cell, that we just covered in cell. So some of the sugars that go into aerobic respiration, those just got made from photosynthesis. Some of them, again, are going to be shipped elsewhere in the plant, like down to the roots, or into the stem, or into floral tissues. And some of the sugars are used then to build with. So build what?
Cellulose.
Cellulose would be one. The sugar parts of nucleotides as well. Anything that uses sugars as a building block. So sugars become available by photosynthesis, and then they will be traveling to different locations depending on the plant's moment-to-moment needs. But sucrose is the dominant sugar that's moving through the plant. And we're going to come back and see that next week when we talk about literally how do they move from leaf downward. That's a cool process. If the whole plant together is in a positive sugar balance, so conditions are good, it's summer, it's photosynthesizing, it has all the nutrients that it needs, and it has sugars very much in excess, it can store those as starch. So this would be a 4th location. So ship sugars for immediate use by other tissues. Use some sugars to build, use some sugars in the photosynthetic tissues to do aerobic respiration, or put the extra into starch. And so the starch, if a plant is making starch, it's going to end up in tubers like potatoes, which is my favorite form of starch. Where else can a plant store starch? do they send it? Tubers. Tubers are enlarged starch-filled roots.
Could they send it to the fruit, maybe? Seeds?
Seeds are the other big one. So seeds within the fruit, if it's a fruiting plant. But if we think about what our sources of starch are that we take from the plant, potatoes, sweet potatoes, sugar beets, and then everything else that we use as grain sources of starch, that's from the plant storing it in the seeds. So rice, super starchy. The point of it was to help feed the embryo coming out of the seed. And we use those for our carbohydrate needs. Because we can digest this, we can't digest cellulose, which is the other major place that the plant is putting sugar to build with. So that's just, I don't want you to get so lost in the details of photosynthesis that you forget what the point of it is. The point of it is to build sugar for plant use. We tend to steal it for our use. But photosynthesis is how the plant is making that. So I'm going to switch slide sets now over to the one that's talking about photorespiration. I've already got that here. Right here. Got it twice. Okay. And this particular topic, if you were not here for Wednesday when we talked about Calvin cycle and especially Rubisco, you'll need to go back and review that for sure. One thing, if you watch the videos, if you had time to watch those videos, did you notice that they don't mention Rubisco in the videos?
Yeah.
That drives me crazy. My Principles of Biology book that I use also does not have within the whole book any mention of Rubisco, which is also just crazy. So I'm still going to teach about it. Because Rubisco is the reason why photorespiration happens and the reason why carbon fixation happens. So this lecture is based on actions of Rubisco. So like we talked about last time, Rubisco has two actions. So we've got ribulose 1,5-bisphosphate. That's the rubis part. And this enzyme Rubisco is combining a 5-carbon molecule with either CO2, and that's the C designation in Rubisco. And this is the intended substrate. This is what Rubisco originally evolved to do, is to put these two together. But we talked about last time how this molecule and that molecule are chemically similar enough that oxygen can bind in the binding site for CO2. It's shorter, but it's got the same general character on each end of the molecule. So if Rubisco is binding oxygen, and that's the O part in its name, it can add oxygen onto ribulose 1,5-bisphosphate as well. So that's why the whole enzyme is ribulose 1,5-bisphosphate carboxylase oxygenase. It can do either. Which one works better generally? Which one does Rubisco usually do?
CO2.
Yeah, so it binds CO2 much better than oxygen. So that there's a competition in the binding site between CO2 and between oxygen. CO2 will win, usually. So if Rubisco is fixing carbon, CO2, it makes a 6-carbon intermediate that immediately falls apart into 2 3-carbon intermediates. And because this is the first prevalent form of fixed carbon, we call this C3 photosynthesis. If Rubisco fixes oxygen, there's no new carbon coming in here. This is like oxygen fixation, not carbon fixation. So if we have oxygen adding on to a 5-carbon molecule, you get one of the correct thing, 3-phosphoglycerate, same as up here. But then you get this other 2-carbon molecule. And this one is not very usable by the plant. We formed something. We have not fixed carbon. This is not something that's very valuable for the plant. So if this is one of the byproducts, this is a problem for the plant. And the reason that it's a problem is that the plant must regenerate that back into the Calvin cycle some way. That's what the next slide is going to be about. What happens to 2-phosphoglycolate in that situation? Okay, so here's 2-phosphoglycolate. That's what has been made, unfortunately, if Rubisco fixes oxygen. So it's made in the stroma. That's where Calvin cycle is happening. So we end up with this 2-phosphoglycolate, 2-carbon molecule in the stroma. The plant will eventually convert this back into the correct molecule. Eventually. Into a 3-carbon molecule. So to do that, it has to leave the chloroplast, go into the cytosol, enter the peroxisome, and in some of these stages it's undergoing chemical reactions along the way. But the path of the intermediates, chloroplast, cytoplasm, peroxisome, cytoplasm, mitochondrion. An intermediate comes back to the cytoplasm, back to the peroxisome. It's ridiculous. And every time the intermediate crosses a membrane, there has to be some energy used to get it across the membrane. So all of this, the reason that I put this slide up here is for you to just remember that all of this takes energy. Doing photorespiration is an energy drain on the plant. It's not just that it's not fixing carbon, but it takes energy to return this compound back to what can actually go through the Calvin cycle. So it takes energy, and it definitely also reduces carbon. Is that cable or carbon? It reduces carbon fixation. And this process is called photorespiration. The reason it's called that is because it was discovered that it happens before researchers knew why. So they knew that if they shine light in dry conditions, plants are releasing CO2 and using up oxygen, like we do when we breathe. We release CO2, we use up oxygen. That happens during this process when the lights are on. So they called it photorespiration. That's where the name comes from. So when this happens, this is a huge problem for plants. And this tells you when is it likely to happen. So maybe add on here, no, it's already on there. What conditions promote this? So what we're looking at is the bottom side of a leaf with stomates open or closed. The blue arrows are CO2. My kids are on the other side of this wall. You just came in. That's what that like super excited sounds are from that side of the wall. Blue arrows are CO2. The red arrows are how much oxygen is being produced during photosynthesis. Where's the oxygen being produced? What part of photosynthesis?
The light reactions.
Yeah, so the light reactions are kicking out oxygen because they're splitting water. The Calvin cycle is sucking in CO2 to fix it. So we've got CO2 coming in, oxygen going out. So in these pictures, the fatness of the arrow tells you how much of that substance. So if on a normal day, when the stomates are open, we've got CO2 diffusing into the plant, oxygen diffusing out of the plant. And there's more CO2 than oxygen inside this space. That's actually not true. These represent how much of the thing is Rubisco binding, not the absolute amount. So in this case, Rubisco is binding CO2 mostly. If the stomates shut, there are no gases moving across that membrane because of the cuticle. CO2 gets used up. So whatever CO2 is in there, it will get used up in the Calvin cycle. Oxygen can't leave. So the oxygen builds up. And that shifts the balance between how much CO2 is there versus oxygen. And so in that case, the levels of CO2 binding are about equal to oxygen binding. And the plant will start to do more photorespiration. Starts to waste energy. If that still continues, so this situation still shut, CO2 is used up even more. And oxygen is accumulating or building up even more. The plant overall will start to lose, either not fix any carbon or even possibly lose carbon from itself. Very dire for the plant. So what would make the stomate shut then, if this is so bad for the plant? Drought. So if the plant is losing too much water, water is more important to the plant moment to moment than fixing carbon. So this is a short-term choice to conserve water, but it tends to cause photorespiration to happen. Yeah.
How does the plant lose CO2 if the stomate is closed?
That's a good question. So where it's losing, the question is, how does the plant lose CO2 if the stomates are closed? It can build up in the airspace. Like it can build up in the airspace, which is not technically in the plant, if that makes sense. But thinking of the airspace as a separate compartment is useful to think about these. So we think, once something goes in the stomate, it's in the plant. Well, it's in the air. Once it's in the cells, it's in the plant. Yeah, that's a good question. So plants would, doing photorespiration is a problem for plants. Some plants, which we call C3 plants, they're stuck with this. If their stomate's closed, they're going to photorespirate. They may not be able to assimilate carbon. Some plants, over time, have evolved the ability to do an alternative type of carbon fixation, and we call this C4 photosynthesis. Please note that these plants also can do C3 if it's not too dry. So they would still prefer to do C3 if conditions are good, but in dry conditions, they can switch on this alternative style of photosynthesis and not suffer as much photorespiration. And we call these C4 plants. One thing that you should memorize are what are the major C4 plants. This comes up on standardized tests. Like, this is one of the most important things that biology majors need to memorize. It's not, but it appears there. So, what are some C4 plants?
Corn.
Corn. The American C4 plant. What else?
Rice.
I don't think rice is.
Doesn't rice grow in water?
It does.
I don't know why that
I had never thought about that, but that makes sense that rice does grow in water, and that's probably why it's not a C4 plant, although it is a grain. It's a good guess. These other ones we're not. I should have written corn here. This is lame. Right, this is corn. This kind of looks like the stem of corn. What are the others?
Wheat?
Wheat is not.
Is that sugar cane?
I think it is sugar cane. It looks like corn-like stem. What about this one? I think this is sorghum. Sorghum is a C4 plant. And the other one that you should probably go ahead and learn is millet. So we know corn because that's the American grain. Sugarcane, sorghum, and millet are other C4 plants. Those are not the only C4 plants, but those are the ones that tend to appear on standardized tests and are like the worldwide food crops that are C4. So these can do C3, but in conditions where it's needed, they can do C4 photosynthesis. Checking for time here. I think I moved a slide when I was prepping for this lecture. So if your slide packet skipped one, it's in there later. This leaf anatomy difference between C3 and C4, I remember covering this at some point when I was in general biology, but I did not understand it at all. So if it feels like I'm going over this too much, it's because of that. This is a C3 leaf, and this is the one that's similar to the one I had you learn the anatomy of in the anatomy section. So it's got epidermis up here, cuticle all over, guard cells down here. But the main thing that tells you that this is a C3 leaf is how the vasculature or the vascular bundles are arranged. What's in the vasculature? Xylem and phloem. So water and sugar carrying elements. And surrounding those are the mesophyll. So we talked about before we have palisade mesophyll, and then we have spongy mesophyll. And the reason the spongy is because it's got air pockets all around it. So a C3 leaf has relatively more air space in it. And the air space and what that touches matters a lot to how this operates. What part of this leaf is doing photosynthesis?
Palisade mesophyll.
Both the palisade and the spongy as well. So all the mesophyll is doing photosynthesis. And if you let your eyes relax and you look at where the air space is, it's definitely amongst the spongy. It's also reaching up near the palisade mesophyll. So in the C3 leaf, the mesophyll cells are near the air. And when they're near the air, they are near to CO2 and oxygen, both. And the vascular bundles are kind of dispersed throughout. That's C3. Now C4, this, when I saw these in a lab at some point on a microscope slide, I decided that they looked like monkey faces to me. They looked like a face. And I remember seeing that, but that's like all I remembered about it. Like that was it. It's like, oh yeah, that's one of those monkey face leaves. This is a C4 leaf. So a leaf that is able to do C4 as well as C3. It has different vascular bundles. And the next time that I saw one of these monkey face leaves was when I did my biology licensure exam to teach high school. So I'm telling you now, this was on there in a black and white drawing, these monkey face leaves. And I think it was asking the question and the answer was C4. And I managed to like pull that out of there. I was like, I think it's C4. This is a C4 leaf. So the monkey faces are the vascular bundles here. And they are much larger and much more interesting looking. Notice also that there's very little air space in this leaf. So it makes me wonder if I took like a cross section, certain area of C4 leaf and weighed it next to a C3 leaf, I think this one would weigh more because there's less air, more material. So we have less airspace to start with, and we have, let's say, more complex vascular bundles. And the next picture is going to be a cartoon that shows what kind of cells are actually in those vascular bundles.
This is kind of a random question, but do you think that it's possible that, like, for example, with corn, has it always been C4, or do you think it, like, developed to be?
It definitely, so this is a thing that Han would know, because he literally studied grass evolution, which corn is. One of the ancestors, if you go back far enough, would not have had C4. And people definitely know when in the corn ancestry you can identify C4 characteristics. I don't know,
Yeah.
but people do. The other interesting thing is that in different plant families, C4 has developed more than once, which is amazing to me. Like it wasn't just like suddenly on Earth there's the appearance of C4 and then all of the ones that we have now are descended from that. It evolved independently. You could probably, I mean, you're familiar with that concept of independent because you had the class. Like convergent evolution is what it's called, yeah. Convergent evolution of traits. I don't know, but it's a very well studied topic by other people. All right, so here's the cartoon of a C4 leaf. There is airspace. It's not that there's no airspace, but there's less airspace. And there is this radial arrangement of mesophyll cells. So we don't really have palisade or spongy in a C4 leaf. It's mesophyll around the vascular bundles. And the most special thing about C4 plants is that they have these cells called bundle sheath cells. So bundle for the vascular bundle, which would be in here. Sheath as in it's like totally going around it and encircling it. Bundle sheath cells. And the existence of these bundle sheath cells is what makes C4 photosynthesis actually work. It's a structural characteristic. So here are the regular leaf mesophyll outside the bundle sheath. And they are next to CO2 and they are next to oxygen. Where the air is, CO2 and oxygen are in those air space because they came in through the stomates, which in this picture are closed. The bundle sheath cells are farther from the CO2 and farther from, or further from the oxygen. So if you imagine CO2 and oxygen, they will diffuse from air to water And there are like physical laws of physics and chemistry that describe that. Gas equilibrium stuff. They will diffuse into this cell. They could eventually diffuse into this cell, but it's far enough away that not as much will make it there. And this is why, like in our bodies, we have so many capillaries to get oxygen and CO2 close to cells and have it a diffusible distance. These gases diffuse through cells, but it takes them longer to go to extreme depths. And the further away that this is from the airspace, the less these are diffusing in, is what I'm trying to say. So, in C4 plants, they would prefer for Rubisco to work in here more than out here. This is a strange statement.
For where?
In the bundle sheath cells. That's, yeah, that's important. Here, when I say here is the bundle sheath cells. There is Rubisco out here. There is Rubisco in here, but the Rubisco that's operating in here is not coming into contact with as much oxygen. It's also not coming into contact with as much CO2. So the trick of C4 photosynthesis is to get CO2 and not oxygen from the air in here for Rubisco to work on it. And that's what we're going to see next, how that actually happens. So maybe add on to this picture one more thing with the red marker. So in the mesophyll cells, those would be doing more photorespiration, but inside the bundle sheath, those cells would be doing less photorespiration. And the next couple of slides are going to explain why. So this picture and the next are the most important ones in this lecture and on this topic. And what they're showing in the gray outlines, here's the lower leaf epidermis, here's the stomate, this is the regular mesophyll cell, this is the bundle sheath cell, and then up here would be the xylem and the phloem. So we're looking at compartmentalization. And what C4 plants do is the CO2 that diffuses into the mesophyll cell the plant is using an enzyme that's not Rubisco. There's a separate enzyme in these cells, not Rubisco, that combines CO2 with phosphoenol pyruvate, same one that we saw in glycolysis if you're in cell. On those videos, they are saying that, both of them, strangely enough, were saying that PEP was phenylenol pyruvate. It's phosphoenol pyruvate. I feel like one of them watched the other's video and then repeated the same mistake. Still phosphoenol pyruvate, which is a 3-carbon molecule. And that's combining with CO2, which is a 1-carbon molecule, to give oxaloacetate. Where did we see that recently?
Citric acid cycle.
Yeah, it's part of the citric acid cycle. So, same chemical, except now it's out in the cytoplasm. And because the first form of fixed carbon is a 4-carbon molecule, that's why this is called C4. So the photosynthesis styles are named after what's the first form of fixed carbon that the plant gets. In this case, it's oxaloacetate, and that's a 4-carbon molecule, so C4 photosynthesis. The oxaloacetate can move over into the bundle sheath cells through these holes, which are what? What are those holes? It's been a while. Plasmodesmata. This is small. This can fit through plasmodesmata. You know what can't fit through plasmodesmata? Enzymes, like Rubisco, and this enzyme, which has a name. It's PEP carboxylase, I just remembered. PEP carboxylase. You don't have to know that. But oxaloacetate, or in some cases malate, which is also a 4-carbon molecule, can fit through the plasmodesmata, and when it arrives in the bundle sheath cells, it drops off CO2 and reconverts into pyruvate, which is a 3-carbon. So in the bundle sheath cells, CO2 is enriched in there, and that's what these numbers are referring to. So the CO2 concentration in the bundle sheath cell is 70 micromolar in this scenario. In the mesophyll, it's 5 micromolar. And in the airspace, it's 150 parts per million. Why doesn't it match?
It's a lot smaller.
Because this is a gas in gas measurement. This is a in water measurement. It's annoying. But that's why the different units. So this level what's in here is lower than both of these, because there's a mechanism that's moving CO2, scavenging it out of here up into here, and so this value is actually more than 5 micromolar. So what the plant is doing here is it's capturing carbon here, just not with Rubisco. It's grabbing it temporarily onto a 4-carbon molecule. That travels into the bundle sheath cells and drops off CO2, and in fact enriches this for CO2. So the Rubisco that's up here, there's a lot of CO2, there's not much oxygen. There's some Rubisco down here, there's some oxygen down here, but the Rubisco that's in the bundle sheath cells will definitely preferentially fix CO2 because its concentrations are artificially kept high up here. Does that make sense? Like if I'm Rubisco and I'm down here, it's kind of equal whether I'm going to grab oxygen or CO2 when the stomates are closed. If I'm up here and I'm Rubisco, I'm much more likely to grab CO2 because there's more of it around because of this shuttle system. And that's how C4 photosynthesis works.
So the enzyme that gets the CO2 in the mesophyll wouldn't grab oxygen as well?
It doesn't.
At all?
It doesn't, which is a benefit. So the question was, the enzyme that grabs CO2 in the mesophyll cell, does it grab oxygen also? And it doesn't. So that's also key to making this work, is that it's grabbing CO2, putting it into a form that can be delivered to the bundle sheath cell, but oxygen can't take this ride. That's important. So it doesn't have that drawback of Rubisco. If you ever get, you probably won't. I'm trying to think if any of you will ever have this kind of question on one of your professional tests, and you won't, unless you're going into plant biology, but this is a big deal. How does the structure of a C4 plant allow it to preferentially fix carbon, or to inhibit photorespiration, or to inhibit Rubisco from binding oxygen? It's a structure function. structure, function, thing. What's close to the air? Where is CO2 enriched? That's something to focus on for sure. Okay. I forgot my watch today. I have to keep checking the time. One question that sometimes comes up is, all right, well, if photorespiration is so bad for plants, why haven't they selected it out? Like if this is a trait that is detrimental, why haven't plants developed, say, forms of rubisco that cannot do photorespiration, that can't bind oxygen? And that's a totally valid question. And this is related to the question that someone asked last time about why would rubisco ever bind oxygen in the first place. So remember that rubisco, I'm going to switch to red, this one's drying out. Rubisco evolved before oxygen was prevalent. So if you think back to when we talked about early plants and how they were derived from early photosynthesizers, algae, and then before that, photosynthesizing bacteria, rubisco was around before there was oxygen in the air. So if a molecule evolves to bind CO2, there's no problem for millions and billions of years. There's no oxygen. Oxygen comes along. It's smaller than CO2. I mean, you can't make the binding site not fit oxygen, then it wouldn't fit CO2. And the answer to this is it seems like rubisco can't evolve in that way. So think about this. If this is rubisco, this is one of the subunits, and that's the binding site for CO2, right in there. How could it change to let CO2 in and not oxygen? That's very difficult, because they're both so small. They're too chemically similar. And actually, scientists have attempted to take rubisco and artificially create a version that won't do photorespiration. It's just not working. Why would scientists want to do that? That's so dorky. Why would you make that your life's work? Or your 20 years' work?
Plants could grow faster with less. They wouldn't have to worry about losing water. No, that's not true. They'd be more efficient, at least I guess, because they wouldn't waste all the energy fixing oxygen as well. So, like a crop could grow faster. Yeah, that's the word.
You're not from an ag background. But what you're saying is correct. So if you could make rubisco in the world stop photorespirating, and that'd be great. Plants would yield better. They could be more drought tolerant, because at some point, if you have to keep your stomates closed because of drought, the photorespiration will sap your resources. So yield, drought tolerance. I mean, it was such a great idea. As soon as someone thought of this, yes, we need to change plants in this way. And so they did directed mutagenesis and trying to change rubisco to make it not bind oxygen and only bind CO2, and it was pretty much a resounding, disappointing failure. So it seems that, I mean, it could be in millions of years that some plant will naturally develop that type of mutation, but not so far. And I left hanging all this time, what is going on with these plants? I don't even remember now, honestly. So don't focus too much on this. There was something about this plant that it made it do more photorespiration than usual. But I don't remember now what it was, so don't worry about this too much. Also, this slide, when I was going through today, I could not remember what the point of this was. It's probably related to the previous slide, so skip over that for this year. So there are other plants that grow in especially dry conditions that are C4 plants, and they've evolved. I mean, it's obviously, it's easier to evolve a whole photorespiration tolerant system in C4 photosynthesis than it is to evolve a rubisco that doesn't photorespirate. Because this has happened multiple times. A better version of rubisco has not really happened in billions of years. Like, it's become 8-fold rubisco in plants, but it's not hugely different than what exists in bacteria. Okay, perfect. So on Monday, I'll be talking about CAM, cryolation acid metabolism, style of photosynthesis, and then also a little bit of material about sucrose transport. So once a plant makes sugar, maybe it's had to overcome photorespiration, maybe not. How does it move the sugar? And so those topics will be Monday. I think we have a review day Wednesday, exam on Friday. Is that right? We don't have lab on Monday. So remember, we don't have lab. Some of you should go to advising. Some of you should go to the seminar makeup session interview. I think some of you already said you're coming to that. So we will have class, we won't have lab.